In 1987, Atsuo Nakata and his team of researchers from the Osaka University in Japan first reported the presence of clustered, regularly interspaced, short palindromic repeats, abbreviated as CRISPR, in the Escherichia genome. The clustered, regularly interspaced short palindromic repeats refer to short and interrupted repeat sequences of DNA nucleotides found within the genome of prokaryotes. These sequences are the same when read from 5' to 3' on one strand of DNA and from 3' to 5' on the other, i.e., the complementary strand, and are hence termed as palindromic repeats. Thereafter, its presence was further reported in both gram-positive and gram-negative bacteria, along with archaea. This led to the obvious question regarding the importance of CRISPR. Much later, in about mid-2000s, the functionality and importance of CRISPR was first realized in prokaryotes. The CRISPR system is a key component of their adaptive immunity that protects these prokaryotes from the attack of viral DNAs, bacteriophages, and plasmids. Adaptive immunity refers to the immunity that an organism acquires after exposure to an antigen, either from a pathogen or vaccination. Vaccination, for example, is a form of adaptive immunity since the body is exposed to antigens and form antibodies in response, which contributes to the immunity developed. Although initially revealed to be a genomic component of bacteria and archaea, CRISPR has found its way through into genome editing in various eukaryotic species. In this presentation, I would be first describing functioning of CRISPR technology as a defense mechanism in prokaryotes and thereafter, the molecular pathway it undertakes during genome editing in humans and other organisms. When placed under an attack by virus or phage, the bacteria signals its immune system to get activated to defend itself from the invaders. The entire process of acquiring immunity can be categorized into 3 key steps: acquiring repeating nucleotide sequences, also known as short palindromic repeats, CRISPR RNA, abbreviated as CR-RNA, biogenesis, and the interception of the target. All these steps will now be explained in details. The first step involves acquiring the repeated nucleotide sequences by the host from the invader. For this, a trans-activating CRISPR RNA, abbreviated as tracker RNA, is needed. The term trans-activating here implies that the CRISPR RNA and the invading target DNA are different molecular entities, and one acts upon the other. In order to obtain the repeated nucleotide sequences from the target DNA, the host needs tracker RNA along with a CRISPR-associated, also called Cas nuclease protein. A nuclease protein is an enzyme capable of cleaving DNA by nucleotide linkages, just like a scissor. Cas9 is one of the CRISPR nucleases found in Streptococcus pyogenes. While this bacteria is a pathogen towards humans and can cause several diseases such as pharyngitis, tonsillitis, scarlet fever, and many others, interestingly, it knows how to protect itself from other attacking viruses and phages through its CRISPR Cas9 system. Cas9 nuclease is one of the most extensively researched and characterized CRISPR-associated nuclease protein, and I will be referring to it to exemplify the mechanism of CRISPR. So, basically, the tracker RNA, along with the Cas9 protein, initially selects the repeated nucleotide sequences within the target DNA of the invading virus or phage. The repeated nucleotide sequences, or the short palindromic repeats, are alternately interrupted by nucleotide sequences known as protospacers. It is to be noted that the selection of the repeated nucleotide sequences is, however, not a random process, but is guided by the protospacer adjacent motif, abbreviated as the PAM. The PAM is a small 2 to 6 base pair DNA sequence that lies adjacent to the site where the cleavage is supposed to occur and helps in guiding the Cas9 or the scissor to the site of incision. In fact, the site of cleavage of DNA nucleotide sequence is situated 3 base pairs upstream of the PAM region of target DNA. The Cas9 nuclease has 2 domains, the HNH and the RUVC domains, joined by a linker. Upon selection of the target DNA sequence, the Cas9 nuclease binds to the target in the form of an R loop, and the conformation actually resembles the alphabet R in 3 dimension. The Cas9 nuclease thereafter undergoes conformational change in order to bring its 2 domains, i.e. HNH and RUVC, in contact with the target DNA strand to initiate the cleavage, and a series of cuts are made in a scissoring fashion. So basically, the Cas9 nuclease is scissor in CRISPR toolkit. The next step involves biogenesis of cRNA, i.e. the synthesis of CRISPR RNA, cRNA. In this step, the repeated nucleotide sequences acquired from the target DNA of the invader is incorporated into the bacterial host genome. This step is crucial in acquiring the adaptive immunity, since it creates a memory of the viral or phage invasion which comes to rescue when re-attacked by the virus or the phage. The acquired nucleotide repeats are woven into a CRISPR array within the host genome. This array is then transcribed or arranged into a 20-nucleotide sequence called pre-CRRNA. This pre-CRRNA will obviously have complementary sequence to the tracker RNA, which helped in its recognition in the first step of spacer acquisition through complementary base pairing. The pre-CRRNA joins with the tracker RNA through complementary base pairing in presence of RNase 3, which is an enzyme capable of cleaving double-stranded RNA, and other factors to form a duplex structure, also referred to as the tracker RNA CRRNA complex or the single guide RNA. The term guide is synonymous with the role of the single guide RNA, since this RNA guides the nuclease, i.e. Cas9, to the site where the DNA is cleaved. The final step involves the interception of blocking a viral or phage attack in future. Under a repeated attack, the sgRNA complexes with the Cas9 and activates it, forming the sgRNA Cas9 ribonucleoprotein complex, and guides it to the target DNA sequence of the intruder. As we have seen during the first step of acquisition of repeated nucleotide sequences, in the final step too, the target sequence recognition is determined by PAM. When the complementary sequence is identified by the sgRNA, the sgRNA Cas9 ribonucleoprotein complex binds to the target DNA, and the latter is cleaved, rendering it ineffective, and the intruder is killed. In 2012, Jennifer Doudna, a molecular biologist from the University of California, Berkeley, along with French microbiologist Emmanuelle Charpentier, were the first to propose that bacterial CRISPR Cas9 could be used as a programmable toolkit for genome editing in human and other animal models. This brings us to another interesting section of the presentation, where I will discuss the use of CRISPR to manipulate the genome of humans and other animals. Genome editing using this technology can be achieved by inserting nuclease protein such as Cas9, which is usually sourced from Streptococcus pyogenes, with an engineered and complementary sequence of sgRNA with respect to the target DNA. Upon introducing these components into the cell where genome editing is desired, the sgRNA activates the Cas9 nuclease and also binds to the target double-stranded DNA, abbreviated as dsDNA, through complementary base pairing. The nuclease protein, i.e. Cas9, thereafter cuts the selected dsDNA sequence and thereafter gets detached from the target dsDNA. While the HNH domain of Cas9 cleaves the target DNA strand with complementary sequence of sgRNA, the RUVC domain cleaves the other strand of the dsDNA, i.e. the non-complementary strand. After the incision is made, the natural DNA repair mechanism sets in within the target DNA. The cleaved dsDNA can either undergo repair via 2 routes, i.e. either by homology-directed repair, HDR, or non-homologous end joining, NHEJ. The NHEJ pathway repairs double-stranded breaks in DNA by directly ligating without the need for a homologous template, i.e. a DNA strand with similar sequence that can act as a template. The NHEJ mechanism can also introduce insertion or deletion of specific sequences at the joining ends, thus creating indels. Indels are DNA strand with insertion or deletion of nucleotide sequences. Thus, NHEJ produces DNA strands with non-uniformity in size. The other route of repair, i.e. the HDR pathway, is commonly found in bacterial and archaeal cells, while the former, i.e. the NHEJ, is more common in eukaryotic domain. The HDR process, although relatively complex than NHEJ, uses homologous DNA template. The homologous DNA template has homology to the adjacent sequences surrounding the site of cleavage to incorporate new DNA fragments. The template guides the repair process, and it lowers the possibility of errors. And since there is not insertion or deletion of nucleotide sequences, HDR pathway maintains uniformity in the size of the resulting dsDNA, unlike NHEJ. So the molecular mechanism of CRISPR genome editing technology sounds really simple, right? Now we move on to the potential applications of CRISPR technology, which has only widened ever since Jennifer Doudna and Emmanuelle Charpentier emphasized on the possibility of using CRISPR for genome editing in humans and other animals. The potential scope of application of CRISPR includes its use as a genetic screen to identify genes in different cells. One of the prominent applications is in cancer immunotherapy. In this therapy, the immune T cells that are a type of white blood cells that fights against a disease are genetically modified using CRISPR technology. In cancer immunotherapy, these T cells are extracted from the patient's body, are genetically modified to make them more specialized in recognizing cancer cells, and killing them after being reintroduced into the patient's body. Similarly, CRISPR has also found its application in therapeutic management of acquired immunodeficiency syndrome, AIDS, caused by human immunodeficiency virus, HIV. Conventional antiretroviral therapies are capable of suppressing viral replication. But once the virus gets converted to its proviral form, conventional therapies are ineffective in targeting the virus. The provirus resides within the immune cells and continues to make copies of themselves using the immune cell machinery, and the immune cells fail to target the proviral latent reservoir, which presents the risk of viral rebound or relapse of the disease. Other than cancer and AIDS, CRISPR has also found immense application in developing assays to detect SARS-CoV-2 infection, the cause of the global pandemic. Although genome editing of human embryos and their implantation into human womb and genetic editing of somatic cells have wide ethical concerns and potential risks, CRISPR holds promise to cure various diseases and prevent inheritance of gene-linked diseases. Additionally, genome editing in plants using CRISPR introduces the scope of making plants resistant to certain diseases, improving their phenotype, i.e., observable characteristics, incorporating certain specific traits, improving crop yield, etc. So guys, that is it for this video, and I hope it was useful.